infectious diseases, research, medicine, health. Welcome to Outbreak News Interviews. And now, broadcasting from the Outbreak News Skylar Studios in beautiful West Central Florida, here is your host, microbiologist and editor of OutbreakNewsToday.com, Robert Harriman.
Well, hey everybody, this is Robert, and welcome to Outbreak News Interviews. Now, the NIH awarded a one-year Phase 1 Small Business Innovative Research Grant for $225,000 in funding to Enable Biosciences to develop an improved Lyme disease test, something that everybody's looking for. Columbia University and Tulane University are sub-awardees of this grant. So joining me today to discuss this work is Monica Embers, Ph.D. Dr. Embers is an assistant professor of bacteriology and parasitology at Tulane University. Dr. Embers, welcome back to the show, ma'am.
Thank you.
Okay, before I get into the work that you are doing and your colleagues on with Enable to develop these new tests, I want to ask you about what's the current available diagnostics out there, and one in particular, of course, is the two-step process recommended by the CDC. And there's been a lot of criticism of this over time, and this, of course, includes the EIA test and the Western blot. Dr. Embers, what are the problems with this diagnostic process, and are these criticisms fair?
Sure, I do believe that they're fair. What you're referring to is the current gold standard, which is called the two-tier test, which is recommended by the CDC. And this does include an EIA test or an ELISA and a confirmatory Western blot test. Now, this two-tier test was adopted in 1994, and it's based on what I would consider to be fairly antiquated technology. The ELISA test tends to be fairly sensitive at different phases of disease, but the Western blot is very insensitive, and it's also difficult to interpret. So, all in all, the testing of Lyme disease tends to vary based on what phase of disease. So the real goal here is to be able to diagnose patients at all phases of disease, and especially where two-tier does not do well is in the early phase, where it's only about 40% sensitive, and overall it's about 60% sensitive. So the goal would be to enhance the sensitivity of the assay and also be able to diagnose patients at all phases of the disease.
Now, what other technologies are out there, whether in the pipeline or on the market, and what are the pros and cons as far as you can see?
So, surprisingly, in contrast to other bacterial infections, what we'd like to be able to do is directly detect the pathogen. But the Lyme disease bacterium and the genomic materials of the bacterium and their antigens are not particularly useful for early diagnosis. And this is due to the extreme scarcity of the Lyme bacteria in the circulation. Once it enters the circulation, then it quickly disseminates into the different tissues. And so antigen or pathogen detection, direct detection, becomes very difficult. So there are a number of different platforms that are at or near marketability, including things like microchip-based test, which is available in Europe, and there's another European test based on Luminex, the new Luminex platform, which is a cytometric bead-based platform, and that's what we started our own assay development with. And so what we actually did was we looked at the changes in antibody titer to different antigens of Borrelia, the Lyme disease pathogen, over time. And we saw that depending on what antigen you looked at, you would see a different kinetics. And so some antibody responses would come up early, some would come up later and then stay high. And so by doing this, we were able to come up with a combination of antigens that could be used at all phases of disease. And this is the basis for our collaboration with Enable.
Okay, and the test that Enable's working on, along with you and your colleague from Columbia, is called the antibody detection by agglutination PCR technology. Can you go into that in any more detail?
Certainly. So this platform was developed by Enable Biosciences, and although the techniques themselves are somewhat proprietary, what they do is they link a DNA conjugate to the antigen and use PCR, or polymerase chain reaction, to amplify the signal of a specific antigen-antibody binding event. And so by doing that, you significantly enhance the sensitivity of the detection of antibodies. And this is really important if you have very few antibodies from an antigen that's produced early in infection, then you can detect that. And in some cases, they found sensitivity to be increased over 1,000-fold.
That's great. So what role will you and your collaborator at Columbia be taking on this project?
So, like I said, we developed the or we identified the different antigens that could be used to detect the infection at different phases of disease based on our studies in animals. And so we're essentially producing those antigens to be used in Enable's ADAP platform to enhance the sensitivity of detection of Lyme-specific antibodies, and also we'll be producing other antigens that will target other potential tick-borne pathogens. So we have a more broad spectrum immunoassay that could be used to diagnose both Lyme patients and Lyme patients with co-infections. In terms of Columbia's role, they'll essentially be providing patient samples so that Enable can validate their test with real patient samples.
Gotcha. I mean, for the Lyme sufferer or the future Lyme sufferer that's out there, how long do you think this will be until it's available, if all goes well?
That's a very good question. It takes some time to get from scientific results to an actual product, but Enable Biosciences has a track record of developing tests for HIV and type 1 diabetes, so I think that they have the wherewithal to bring this to market fairly rapidly, given the strength of validation testing, and we're confident that the antigens that we've chosen will be very useful for diagnosing Lyme patients. So, while I can't put a number on it, I think within a few years we should be able to get this out there.
Yeah, and I'm sure there's a lot of people out there that are very excited about this work that you're talking about today. Dr. Embers, any final thoughts on Lyme diagnostics, Lyme disease in general, or anything like that?
Well, as you may know, Lyme disease tends to be fairly controversial, and the way to eliminate the controversy is to arm clinicians with the tests and the treatments that they need to make a reliable diagnosis and to cure their patients. And so with the problems that exist at the two-tier test, I think that if we're able to enhance and improve diagnostics, it's going to go a long way to solve the problems of the controversy and to help patients in the long run.
Yeah, well said. I want to thank you, Dr. Monica Embers, for sharing your time with us today. I appreciate it.
Thanks so much.